Welcome to Mootechie. This slide introduces the world of Archaea by showcasing the wide range of extreme environments they inhabit. These include freezing regions like sea ice, permafrost, and polar areas, deep sea settings such as cold seeps, mud volcanoes, hydrothermal vents, and anoxic zones, and geothermally active sites like hot springs, fumaroles, and hyperacidic lakes. Archaea also thrive in deserts, highly acidic and metal-rich acid mine drainage sites, marine soda lakes, brines, contaminated and nuclear sites, and rock-associated systems formed through serpentinization. Overall, the slide highlights the remarkable adaptability of Archaea across some of the most extreme habitats on Earth. Moving on, microbes of extreme environments, Archaea. It visually represents the concept of exploring where life thrives against the odds. The image likely shows a stark, extreme landscape, perhaps a hydrothermal vent or a salt flat, symbolizing the harsh frontiers where archaea are found. This introduces our journey into understanding these resilient organisms that don't just survive but flourish in conditions most life would find impossible. Moving to our first detailed topic, extreme environments where life pushes limits. Let's look at some key examples. Hot springs and geysers with temperatures ranging from 70 to 122 degrees Celsius and often featuring high acidity or alkalinity, these are like natural pressure cookers where many archaea live. Hydrothermal vents are deep-sea volcanic fissures characterized by high pressure, a lack of oxygen, and plumes rich in metals. They are among the most alien ecosystems on our planet. Hypersaline lakes, such as salt flats, are saturated with brines containing 10 to 35% sodium chloride. Think of places like the Dead Sea. Next are pH extremes spanning from incredibly acidic conditions in acid mine drainage with a pH between 0 and 2 to caustic soda lakes with a pH as high as 10 to 12. Other frontiers include polar deserts, which are extremely cold and dry, the deep subsurface biosphere, miles below the Earth's surface, and zones of high radiation. These environments define the outer edges of the biosphere and are the primary homes for extremophiles. Now, let's answer a fundamental question. What are archaea? We look at the core characteristics of this third domain of life. First, archaea represent a distinct domain separate from bacteria and eukaryotes with unique genetics and biochemistry. This means their evolutionary history and molecular machinery are fundamentally different. Their membranes are built with ether-linked isoprenoid lipids which often form stable single-layer structures called tetraether monolayers. This makes their membranes exceptionally tough. Their cell walls lack peptidoglycan, which is a key component of bacterial cell walls. Instead, they are typically composed of S-layer proteins or a substance called pseudomurane. Their metabolism is incredibly diverse, including unique strategies like methanogenesis, producing methane, sulfur cycling, and chemolithotrophy, which is harvesting energy from inorganic chemicals. Finally, their ecological breadth is vast. They are found everywhere, from the extreme niches we just discussed, like vents and salt lakes, to common everyday environments like ocean waters and soils. Let's categorize these resilient organisms by looking at the main types of extremophiles, each adapted to Earth's harshest frontiers. Thermophiles thrive in extremely hot environments between 45 and 122 degrees Celsius and often featuring high acidity or alkalinity. These are like natural pressure cookers where many archaea live. Examples include sulfolobus and pyrococcus, often found in hot springs and hydrothermal vents. Their proteins and membranes are specially adapted to prevent melting or denaturation. Acidophiles grow best at very low pH, between 0 and 5. Examples are Picrophilus and Ferroplasma, typically inhabiting acid mine drainage and volcanic soils. They maintain a neutral internal pH despite the corrosive surroundings. Halophiles require very high salt concentrations, even up to saturation. Examples are halobacterium and haloferax, which give salt lakes and salterns their vibrant pink and red colors. They manage extreme osmotic pressure. Psychrophiles are adapted to constantly cold environments below 15 degrees Celsius. Example is Methanococoides burtoni, found in Antarctic lakes and the deep ocean. They have evolved antifreeze proteins and more fluid membranes. This slide delves into the specific adaptations to extreme conditions, the molecular strategies for survival. The background image likely depicts deep-sea sediments and trenches, emphasizing these harsh settings. First, unique membranes. The ether-linked isoprenoid lipids and tetraether monolayers drastically reduce membrane permeability and prevent disintegration under thermal or chemical stress. Next, thermostable proteins. These have highly compact structures with increased numbers of salt bridges and ionic bonds for stability. Helper proteins called chaperonins ensured they fold correctly even at high temperatures. Genetic stability is maintained by enzymes like reverse gyrase, which introduces positive supercoils into DNA, making it more heat resistant. Specific DNA binding proteins and modifications to ribosomal RNA also prevent damage. For osmotic balance, halophiles often use a salt-in strategy, accumulating potassium chloride inside their cells to match external salinity. Others synthesize compatible solutes, which are organic molecules that balance pressure without interfering with enzyme function. Finally, specialized metabolism, such as chemolithotrophy, using inorganic chemicals for energy, and anaerobic pathways like methanogenesis, allows Archaea to harvest energy in oxygen-free, mineral-rich niches where other organisms cannot. Now, let's discuss why archaea matter, focusing on their significance in biotechnology and evolution. First, they provide valuable biocatalysts. Thermostable enzymes like the PFU polymerase are crucial for laboratory techniques such as PCR and for various high-temperature industrial processes. In bioremediation, their robust metabolism allows them to degrade pollutants in harsh, contaminated waste streams where other microbes fail. For bioenergy, methanogenic archaea are essential for generating methane in biogas production, turning organic waste into usable fuel. Their study offers profound insights into evolution. The discovery of Asgard archaea, for instance, provides key clues about the origin of complex eukaryotic cells, which include plants, animals, and fungi. Finally, in astrobiology, archaea serve as models for understanding the potential forms life could take on other planets or moons with extreme temperature, pH, or salinity. We hope this overview has illuminated how these tiny organisms shape extreme environments, drive essential global cycles, and hold keys to scientific and technological breakthroughs. Your curiosity is what drives discovery. We look forward to exploring more fascinating topics with you.